So you can imagine, I'm showing a pore in the hydrothermal vent here, and I'm showing the catalytic barrier, the iron sulfides in this barrier in the ancient oceans. And on the outside, we've got hydrogen ions, the protons coming from the oceans, and on the inside, we've got the alkaline hydrothermal fluids, and I'm suggesting that we can get through to this chemistry, the Krebs cycle type chemistry, and to make simple things, the lipids that make up the membranes and amino acids. These are all thermodynamically favored and have all been done quite readily in the lab under these conditions. What happens if you make fatty acids? Well, I imagine that they may just form some kind of protocell inside this. Is very convenient, isn't it? I drew it that way. The kind of protocell inside this vent pore. And now we've got CO2 fixation and the Krebs cycle happening inside the protocell. Now, you can accuse me of being over-imaginative here, and that would be fair enough. You could also say it's a hypothesis, and it's a testable hypothesis, and that would be charitable. The idea is we can make now not just the lipids and the amino acids, but perhaps the sugars and the nucleotides as well, because now we've got an enclosed space, and the thing is presumably the concentrations will be higher, and so on. So it's a hypothesis. Does it work? Well, the first question is, can you make protocells under those kind of conditions? These are quite extreme conditions. I'm saying they're forming on the alkaline side, and the pH there is about 11. Well, it's a simple experiment. We take mixtures of the fatty acids and the long-chain alcohols that have been made under those conditions in the lab previously. It's probably worth saying they've been made in steel reactors, and it didn't work in glass reactors. This is the kind of confounding detail that you often get in science. So we know it kind of can work, but we're not really sure if it really would. But using the fatty acids that were made under those conditions, you can see there. This is cryo-electron microscopy, and those are protocells with a membrane around the edge. This is pH 12, 70 degrees centigrade, modern ocean salinity in the presence of calcium and magnesium ions that normally turn these things to soap. So it works perfectly well. And then you can see with fluorescence microscopy at the right-hand side that we get quite a lot of these, and there's some funky shapes there as well. They're not just all little spheres. Sometimes they're long, extended things, and they move around. I'm only showing you pictures here, but it's quite dynamic. Just heat currents in the environment mean that they fuse together and separate again, so they're quite dynamic. And we were trying to think if we could, what interactions would we get between amino acids and the iron sulfide minerals? Can we get something closer to the 4Fe, 4S clusters? So we tried just mixing them, and it didn't work at all. Just cysteine, which is a sulfur-containing amino acid with iron, and so ferric chloride and sodium sulfide. Mix them together, everything goes black, everything precipitates out. And then we realized, hang on a minute, the only way that cysteine is going to get its sulfur to react is if it's lost the proton. We have to do it in alkaline conditions. So we try it again at pH 9, and suddenly, this is on the left-hand side, UV vis spectroscopy. Suddenly we see a shoulder. You can just see it there. There's a kind of the way that traces down. There's a shoulder there at 420 nanometers. That's diagnostic for 4Fe, 4S clusters. As soon as you see that, you think, whoa, 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 hang on a minute. We are getting the biological clusters that you find in ECH just by mixing a few ions together in alkaline water. And you can't leave it at that. We did a lot of additional work on it, and they really are. You really do. If you mix them up, you get these biological clusters forming spontaneously. And at the right-hand side, you can see they're electrically active as well. They'll pick up electrons, and they pass on those electrons. We haven't made them reduce CO2 yet, but in theory, they ought to do that, and we're trying to do so. I was showing a cluster there as well. That's the shape of these things. They really are like little nanocrystals. So amazingly easy, shockingly easy to make these biological clusters that are responsible for CO2 fixation in nature, just with a single amino acid. I'm showing one other thing here before I go on to information and finish. ATP synthesis. This was from a paper that was just published a few weeks ago. And we're not doing all the steps. We're simply, I mentioned the ATP synthase several times. What the ATP synthase is doing is taking ADP and a phosphate and sticking them together. So now we have 3 phosphate groups in a chain hanging off, and that's what's powering work in biology. It's a weird way of organizing things, isn't it? And so, well, the question is, how would you do this if an ATP synthase is a machine that's coming later, and we still need ATP earlier on? is there a simple way of making it? And what you can see here is there's a very simple 2-carbon molecule called acetyl phosphate, 2 carbons and a phosphate group. In the presence of ferric iron as a catalyst, will give you about a 20% yield of ATP from ADP in water with absolutely nothing else there. And we tried a whole bunch of different catalysts. None of them, the rest of them worked, only ferric iron. We tried at least 10 different metal ions. We tried about 7 or 8 different phosphorylating agents, and none of those worked really either, apart from carbamoyl phosphate a little bit. And then at the right-hand side, we tried, you can make it, oops, we can make ATP, but we wondered, what about the other letters in DNA or RNA? Can we make GTP or CTP or UTP? And the answer was no, only ATP. So ferric iron, acetyl phosphate gives you ATP, but it doesn't give you anything else. And this is slightly crazy, it's slightly scary. It tends to say the reason ATP is the universal energy currency is that it works under prebiotic conditions. It just happens spontaneously that way for reasons that we have some ideas, but it's quite freaky anyway. Right, for the last 5 minutes, I think I have 5 minutes. I'm just going to talk about information. I mentioned already these biophysical patterns in the code, and I wanted to show you that. I'm going to skip over this in the interest of time. This is really just saying, can these protocells make enough nucleotides that you could have random strings of RNA inside them? So if anyone wants to ask questions, I can come back to that later on. But the answer in a word is, yes, it's possible. There are various catalytic feedbacks involving nucleotides, so a form of autocatalysis or just a positive feedback loop that allows protocells to make more of the building blocks for RNA. So that's all that's saying. That's mathematical modeling. This is what I want to tell you about, though. If we just assume that we're right, a bad assumption to make, but if we just assume that really life does start from CO2 and hydrogen under these kind of conditions, and then we look at the patterns in the code and we organize them around autotrophic metabolism, this core of metabolism that I've shown you, you can see this is the first letter. So the code in DNA is a 3-letter code. It's called a codon. So 3 letters encode a single amino acid. And there's quite a cacophony. You could potentially have 64 different amino acids, but in fact we only have 20. And it seems like there's a lot of redundancy and there's a lot of mess in the genetic code, and it's not really clear if it's being optimized or if there's biophysical reactions or whatever's going on. There's also this temporal factor. This is the distance. At the top is CO2, and each of these arrows are going into metabolism. And what you can see is that if the first letter is a G, it's in gray. For whatever reason, the grays are coming earlier. They're coming closer to CO2 fixation. Then comes the adenosine as the second one. So it's an A at the second position. And again, you can see it's structuring. It's close to the and then the C's and the U's come later. So it's not just the number of steps, but there's a structure to that. I'm not going to attempt to explain that structure. I'm not sure I understand that structure, but plainly that's not random organization. There is a relationship between the temporal structure of metabolism and which bases are at the first position of the genetic code. This is the second position, and it's organized around what was at the first position. So at the top left, there's a G at the first position, and then the top right is an A at the first position. And then we're looking at the second position, we're ordering them with hydrophobicity. In other words, for those top 2, you can see there's a very clear relationship that the more hydrophobic the letter that's there, the more hydrophobic the amino acid. There's a clear correlation. So we have a hydrophobic base like A, and it's going to interact with a hydrophobic amino acid like valine, for example. And so there's a clear relationship there. It's much weaker for the later amino acids. The top 2 ones are the first 10 amino acids that you would get in metabolism from CO2 fixation. So again, I can't explain exactly what's going on here, but plainly there is a pattern which is suggesting direct interactions between them. And this is the third position, which is normally considered to be basically redundant. This could be pretty much anything goes at that third position. And all I really want you to take home from this is that is not a random picture. There is order in that picture. And I'm just going to give you one example of it. So just below the midpoint line there, there's 2 blue ones. One of them it says GLU, that's glutamate, and the other one says ASP, that's aspartate. And this is an inverted Koton table. It's the opposite way round to the way it's normally shown, which is why it looks a little bit different if you're familiar with these things. So if you go in, you'll see that the first letter is a G in both cases. The second letter is an A in both cases. I'm struggling to read this myself. Is an A in both cases. And then in the middle, it says R or Y. That's a purine or a pyrimidine. And in effect, that's the distinction between them. And that, at the right-hand side, it depends on size. If there is a purine there, purine's a big, bulky thing. And the anticodon, it then has a pyrimidine, which is much smaller, and the size of the amino acid is selected depending on whether there's a purine or a pyrimidine sitting at that third position. I keep hopping forward for some reason. But that's basically it. Do we know anything about the origin of life? I'd be really interested to hear whether you think after this talk. I would say not much. But we can understand the problem. I cannot say that life definitely started down at the bottom of the oceans or anything like that, but I think using life as a guide begins to structure it so that we can think about the questions, we can do experiments, and we can try to understand intellectually how a sterile, wet, rocky planet can give rise to life and a living planet that we know. We need to know that it's a long continuum. I said it's equivalent to bacteria to dinosaurs, going from prebiotic chemistry to the simplest forms of life. But when I'm saying life, I'm talking about DNA, I'm talking about molecular machines, I'm talking about ribosomes. These are sophisticated machines. We've got to cross that long distance. The proposed steps have to link up. You can't just... The problem with the field at the moment is one bit works in this environment and another bit works in a different environment, and no one says, oh, well, do these two environments ever come together? Is there some way in which they, thinking about autotrophic origins means everything has to happen in the same environment right now. That's a big ask, but it is at least a testable question, and it may be that we can show it, or it may be that we fail to show it, which would be, well, still fun. So we need to demonstrate the viability of each step. I do think, though, that life has been almost a forgotten guide to its own origin. It sounds intrinsically circular, but I think it can tell us a lot. If we can understand the principles, then we can begin to think about, would life elsewhere follow the same principles? Are they there for a reason? Are these strong principles, or did it just happen this way on Earth for limited reasons? I have to say thank you to the people in my lab. Questions like this can kill a career before you've even started. These are big, difficult questions. You could easily go through a 3-4 year PhD without getting any papers. Everything can be negative all the way through, and that is a really bad start in science. So you have to be brave to do this kind of work, and they are doing fantastic work. I only showed you a fraction of the data we've got, but they're really doing beautiful work. And they're holding, this was actually a couple of years ago now, as a birthday present, they're all holding the reverse Krebs cycle. And so I keep having to use this same one. But that's fantastic that they are doing this work and perpetuating these great questions and answers. Thanks to the funders, and thank you very much, and to the Wright Foundation. Thank you.